In this video, we're going to discuss deoxyribonucleic acid, often abbreviated as DNI, along with chromosomes, genes, and genomes. By the end of this explanation, you should be able to clearly describe what each of these biological components are and understand how they relate and interact with one another in the human body. Let us start by discussing DNI, which stands for deoxyribonucleic acid. It is the chemical substance that contains all of our genetic information and instructions. DNI is described as a polymer, meaning that it is made up of many small repeating units that are chemically bonded together in a chain. It is composed of two strands that twist around each other, forming what scientists refer to as a double helix, which is simply a fancy way of saying a double-stranded spiral structure. Now, if we were to extract all the DNI from a single human cell and unwind it into a single long thread, it would stretch out to be more than 2 meters in length. Clearly, this would be far too long to fit into the tiny space of a cell nucleus, so the DNI is very tightly coiled and organized into distinct structures called chromosomes. In total, each human body cell contains 46 chromosomes, which are essentially long molecules of DNI tightly wound around proteins and coiled into a compact shape. However, there are only 23 unique types of chromosomes because we inherit one copy of each type from each of our biological parents. This means we have chromosome pair 1 containing one chromosome from our father and one from our mother, then chromosome pair 2, and so on all the way to chromosome pair 23. Now, the 23rd pair of chromosomes is different because these are the sex chromosomes, which determine our biological sex. Females have 2 X chromosomes, while males have 1 X chromosome and 1 Y chromosome, and this chromosomal arrangement is what defines male and female biologically. One point that often confuses people is that chromosomes are commonly shown in diagrams as X-shaped figures, but this shape only appears just before a cell divides during processes such as mitosis or meiosis. For most of a cell's life cycle, the chromosomes do not look like an X at all, but rather appear as loose thread-like structures within the nucleus. Moving on, a gene is a specific segment of DNI that codes for a particular protein. You can imagine a gene as a small instruction manual located within a chromosome that tells the cell how to assemble a specific protein. These proteins are built from smaller building blocks called amino acids, and although there are only 20 types of amino acids available in the human body, they can be arranged in countless combinations to produce thousands of different proteins, each with a unique function and structure. Therefore, the exact order or sequence of amino acids in a protein is crucial because it determines the protein's shape and role in the cell. Because of this, DNI directly controls which proteins a cell can produce, and this in turn determines what type of cell it becomes. For example, red blood cells need to produce large amounts of the protein hemoglobin, which is responsible for carrying oxygen throughout the body, whereas muscle cells require specific proteins that enable contraction and movement. So in essence, genes play a fundamental role in determining both the structure and function of all living organisms. Lastly, we come to the term genome, which refers to the complete set of genetic material present in an organism. It includes all the DNI found in an organism's cells. My genome, for instance, is the entire collection of genetic instructions that make me who I am. While it would be somewhat different from a stranger's genome, it would be much more similar to my parents' genomes, and in the case of an identical twin, the genomes would be exactly the same. In recent years, scientists have successfully mapped the entire human genome, meaning that they now know the full sequence of DNI letters or bases that make up our genetic code. This incredible scientific achievement has many practical applications. For instance, it enables researchers and medical professionals to identify specific genes that are linked to certain diseases. Sometimes these genes directly cause a disease, in which case the condition is called an inherited disease because the faulty gene is passed down from one or both parents. In other cases, the gene might not cause a disease directly, but could increase a person's susceptibility to developing it. A well-known example is the BARRCI genes, which are linked to a higher risk of developing breast cancer. Regardless of the type of gene being examined, this information allows doctors to provide more accurate diagnoses and choose more effective treatments tailored to an individual's genetic profile. Furthermore, scientists are using genome data to develop improved therapies and targeted interventions that could greatly enhance patient outcomes. Another fascinating use of genome analysis is in tracing the migration patterns of our ancestors. Although all human beings share most of their genomes, small differences between populations can provide clues about where and when different groups of people migrated. For example, genetic studies have helped us understand when early humans left Africa and how they eventually spread to other parts of the world, including North America. These insights contribute not only to medical science, but also to our understanding of human history, evolution, and identity.